mic is working. Can you just check? Oh, it's working now. Okay, great. Okay. Good afternoon, everybody. My name is Anya Kelly, and on behalf of the Physiological Society, I am delighted to introduce the Sharpey-Schafer Prize Lecture.
1, 2, 3.
This lecture is named in honour of Sir Edward Sharpey-Schafer, one of the most important figures in the history of our discipline. Among his major achievements were the invention of the Schafer method of artificial respiration and the co-discovery of adrenaline. He also coined the term endocrine. He was one of the founding members of the Physiological Society in 1876, and he founded the journal Experimental Physiology and was its editor from 1908 until 1933. This prize lecture is a triennial lecture, alternately given by an established physiologist and an early career physiologist. And the recipient that we will hear from today, Dr. Sofia Iris Bibli, is an outstanding early career researcher, working in the area of cysteine metabolism and protein S sulfhydration. Dr. Bibli studied at the University of Athens, completing a degree in pharmacy in 2011 and a PhD in pharmacology in 2016. She was then awarded a European Society of Cardiology research grant that enabled her to join the medical department of the Goethe University in Frankfurt as a post-doctoral fellow. Last year, she was appointed professor at the Goethe University and was awarded an Emmy Noether grant to fund her research on cardiovascular cysteine metabolism. To quote from her prize nomination, "She is an excellent example of a young female physiologist with a solid grounding in redox chemistry that is not afraid of modern technology and at the same time always having translation in mind." It is now my great pleasure to invite her to deliver the Physiological Society Sharpey-Schafer Prize Lecture.
Okay, so this works. It's my great pleasure to be here today and also a great honor for me, and I would like to thank the Society for giving me this opportunity to present this lecture today. So we heard a lot of great talks already during the first 1.5 days of this meeting on different aspects of physiology, and this talk will primarily focus on the vascular physiology. What I will try to do in the next few minutes is introduce you how a single amino acid, just cysteine, and its catabolism can regulate endothelial cell function. So what you can see here in this image is nothing more than a simple staining for the VCAT here in green, which really marks this very unique monolayer cell, this very unique single cell layer, which is termed endothelial cell. And this specific cell layer lines the inner part of the vessels. Now, it has very unique functions. One of them is that it is the single barrier between the bloodstream and the tissues, and it regulates multiple processes, including the transport of nutrients and oxygen from the blood to the tissues. It is widely accepted that when this endothelial cell layer is not working properly, then we have multiple disease development of, for example, diabetic complications or atherosclerosis or even cancer. So for many years, it was very clear that scientists need to work on identifying mechanisms that can regulate and maintain endothelial cell function. And as you can see here, there are multiple microenvironmental and mechanical cues that are regulating endothelial cell fate decisions. One of them, that it is a particular focus of the last 10 years, is how intracellular metabolism can impact the processes of an endothelial cell to transition between the different states, from primarily studied from quiescent and homeostatic endothelium to more proliferative phenotypes. But also, there are some first evidence indicating that when the cell transitions from a quiescent and homeostatic state to a more aged state or differentiation condition, then metabolism also plays a very significant role. So this is what my team and I are interested in, on how, during the transition from development and different disease and aging, these metabolic alterations happening inside the endothelium are affecting not only energy and biomass supplies, but rather the bioavailability of very small signaling molecules that we term modifiers, and just highlight some of them, methylation or acetylation. I'm sure that you have heard about them. So based on that, we formulated a few years ago a hypothesis that these metabolites can regulate the epistates of the endothelium, and by epistates we mean post-translational or post-transcriptional modifications that change the endothelial cell phenotypic barrier and drive endothelial cell fate decisions. Now, how to answer that? What our approach was to start from very well-characterized endothelial cell clusters with known phenotypes, perform a multi-omic analysis on them with a primary focus on metabolism, and subsequently link the changes on these metabolites with the epimodifiers that they generate, and finally attack proteins introducing post-translational modifications, changing their activity, and this could potentially explain the phenotypes that we observe and unravel some novel mechanisms on how to target these phenotypes. So we applied that in 4 different endothelial cell end states: the proliferative endothelium during development, the quiescent homeostatic endothelium, the dysfunctional, and the aged endothelium. And if we just get a snapshot of what is going on metabolically in these cells, then this principal component analysis highlights only one thing, that every single endothelial cell end state is characterized by a unique metabolic profile. Now, if we add a little bit additional information on these datasets with gene expression profiles and some bioinformatics to identify the metabolic trajectories and the pathways that they are altered during these endothelial cell transitions, then we can see that multiple of these pathways are affected. Here in this graph, you see big bubbles that they are nothing more than all the metabolites and the genes that we detect during these endothelial cell transitions, color coding on whether they are downregulated, upregulated, or oxylating, and of course whether they are present in all of these transitions. Now here, what drew our attention was the fact that cysteine was one of the most affected pathways. And we continue to understand how cysteine catabolic pathways might potentially affect endothelial cells. So what our data highlight is that indeed in a proliferative condition here, when the vessel develops, the endothelial cells sprout and proliferate, they utilize high levels of this amino acid. Now, if we enter quiescent and homeostasis, this gets into an intermediate state. It's not lost, but it is still present, but it's quite lost when the endothelial cells develop dysfunctional or AIDS characteristics. So based on that, we question whether this cysteinolytic capacity of the endothelium marks the phenotype of a cell, or it can actually cause the observed phenotypes. Now, this is the pathway that I'm going to talk today, and this is the cysteinolytic pathway, which is utilizing sulfur-containing amino acids, cystathionine and cysteine, with the help of a single enzyme inside the endothelium, which is termed cystathionine gamma lyase, or CSC. What this pathway does is that it fluxes 2 different epimodifiers, one being the carbon source and generates acetyl-CoA, and the second one being the sulfur source and generates hydrogen sulfide. So my talk will be around 3 topics on how this pathway might affect endothelial cell fate transitions, in particular how the carbon sources affect endothelial cell proliferation and identity, how the sulfur sources are important for endothelial cell homeostasis and quiescence, and how the protein itself has an unexpected role that can help us fight endothelial dysfunction during aging. So let's start from the beginning. Initially, what we reasoned is that this acetyl moieties generated from this pathway in a highly cysteinolytic cells, which are the proliferative cells, would potentially work as modifiers of histones, altering the epigenetic landscape of these proliferating cells, subsequently chromatin accessibility control and transcriptional control of the endothelium. Now, to study that, we used the postnatal retina developmental model. Here you see a representative image of how this postnatal retina vasculature looks on postnatal day 6. Isolectin before in red stains the vessel, and indeed in this front region, which are the proliferating and the sprouting cells, you have a heterogeneous expression of the key enzyme that catabolizes this amino acid cysteine, CSC. Now, to test whether this region and this enzyme is responsible to flux the carbons, we FACS sorted these cells in different stages, starting from distinguish between the vascular front and the more quiescent and established on postnatal day 6, and compare them with something more mature on postnatal day 15. And what we did was to perform carbon-13 metabolic flux analysis in these cells. By using carbon-13 uniformly labeled cysteine, we were able to see that 30% of this C2-CoA pool inside this developing vasculature comes from the carbons from cysteine. Of course, cysteine is not alone there. When you add additional carbon sources as the hallmark of endothelial cell proliferation, which is glucose, then you can see that glucose and cysteine together support 100% of the C2-CoA pool in the developing vasculature, and this is a little bit reduced when they enter quiescent or mature phenotypes. This seems to be taken over from fatty acids later on. So this study highlighted that this gradient of cysteinolytic capacity is also supporting the generation of a significant presenter of the C2 modifier inside these endothelial cells. So to test whether this is indeed a CSE-dependent mechanism, we generated endothelial-specific inducible CSE knockout mice. Here you can see in this image that the CSE in green is absent in our knockout model, and we performed the same experiments and identified that indeed the carbons of cysteine can no longer flux to generate pyruvate and C2-CoA when the enzyme was not present, bringing us to the conclusions that when the cells proliferate and sprout, require two different sources of carbons to generate the acetyl molecule. Now, why is that? Why the cell really needs two different carbon sources to generate an acetyl moiety? So we reasoned that this molecule coming from the cysteine, this carbon source, is irreplaceably and systematically modify unique histone lysine residues, which result in a cysteine-dependent chromatin accessibility control and transcriptional control of the endothelium. Now, to answer that, we had to go back in cell culture and use wild-type and CSE endothelial-specific knockout cells, which we treat them with a competitive mixture of either unlabeled cysteine and heavy-labeled glucose, or vice versa, unlabeled glucose and heavy-labeled cysteine, and mimic the environment of the developing vasculature in the PAPs, which is hypoxia with increased growth factors, as for example vascular endothelial growth factor, perform histone extraction and do carbon-13 proteomics. And the results were quite remarkable. Here, this graph highlights that every single carbon can be sensed from different lysine residues. In brief, what happens, what you see here in green, is carbons from cysteine decorating histone 3 lysine 56, 27, and 36. They acetylate this specific 3 lysine residues, while glucose, for example, seems to prefer histone 4 and 2 different lysine residues within the histone 3. And all these are also CSE-dependent. Deletion of the enzyme in the endothelial cells does not allow this flux of this acetyl molecule to decorate these specific 3 lysines of the histone 3. Now, proof of concept, of course, simple Western blot analysis proves that indeed a CSE-dependent lysine residue and a cysteine-dependent lysine residue is lost when we delete the enzyme, something that is not true for a lysine residue, that it is attacked primarily from glucose, concluding that indeed it seems that there is a systematic way of these carbons to modify specific histone lysins. So what do they do? What is the impact of these modifications in the endothelial cell transcriptional program? To answer that, we used ATAC sequencing and RNA sequencing data, and here I will just show some key observations of our studies. So what you can see here is actually chromatin accessibility data summarized, and the taller this peak is, the more accessible the chromatin is, and you can clearly distinguish that the capability of a cell to catabolize this amino acid cysteine and generate this carbon source is linked to the accessibility of the chromatin. The lowest cysteinolytic capacity we have, the closer is the chromatin. When we increase it, then we go in a lower order of chromatin. And there, the regions that we identified to be open or more accessible were involved in angiogenesis, VEGF signaling, and of course endothelial cell commitment. And I would rather say that the most important finding of this study was the fact that the transcriptional factor footprinting motif, the one of the ERG1, which is a key transcriptional factor to regulate endothelial cell decisions, commitment, and also proliferation, was enriched only when these carbons could flux to decorate the histones. Now, how does it look transcriptionally? I will not get into many details, but you would appreciate that these different cell types describe and show a rather moderate transcriptional reprogramming, and every transcript involved there is linked, of course, to angiogenesis and VEGF signaling. And with that, we went over to test whether this is physiological relevant. So the fact that the cysteine carbons are fluxing and are regulating the transcription of the endothelial cell may impact endothelial cell proliferation. And indeed, starting from some cultured experiments in cell culture, what we did was very simply to remove the cysteine from the culture media. I have to mention here that the majority of the media that we use in culture have up to 17 millimolar of this amino acid. And as you can see here, within 3 days after deletion of this media, the cells stop proliferating, they stop growing. And indeed, morphologically, you can see that you don't have any EDU incorporation, while in the normal cysteine-treated cells, you have a significant number of cells still proliferating. The control group looks monolayer with a very nice junctions and appropriate morphology for the endothelial cell layer. However, 72 hours after deletion of the cysteine, these cells look irregular with very enlarged bodies. They still do express weak adherine, but we would rather say that this is not how endothelial cells look like. When we proceed with some more experiments in ex vivo models of sprouting, and this is just an aorta model of sprouting, you can clearly see again that deletion of the amino acid results in a loss of the proliferative capacity of the endothelium. Now, we wanted to translate this information, of course, in vivo, and for that we perform a gain of function experiment in which we tried to boost the system to utilize as much more carbons from cysteine as possible by intravitreally injecting solvent and cysteine in the postnatal day 4 of retinas, and then follow them up for about 24 hours. And what you can see here in the zoom-in images, with selecting before in green staining the vasculature, is that the EDU cells in the vasculature, but also outside of it, respond to this increased level of the amino acid to proliferate much more. And if you continue for another 24 hours, then we have this very impressive finding that nearly the retina that received the cysteine has completed development, and the endothelial cells proliferate much more. Now, if we do the opposite experiment, we stop the flux of these carbons, of course, by knocking out the key enzyme, CSE, and we see the opposite phenotype. So we have an underdeveloped vascular plexus, much less dense, with much reduced proliferative cells, and there are very irregular morphology of the tip cells, and of course loss of philopodia, and these are all quantified here in multiple births of the CSE littermates. So what I've shown you so far is that indeed this pathway is used from proliferating cells to utilize the carbons to decorate particularly histone 3 marks that allow the binding of a key transcriptional factor, which is called ERG1, and drives endothelial cell identity and proliferation. However, this study gave us another very interesting observation. It seems that when you enter quiescent, then the carbons should not be the primary modifier that plays a role, but still our cells in the homeostatic human, middle-aged human, and in the mice do utilize this amino acid and do express the CSE enzyme. So we reason that potentially there is a second modifier, the one from hydrogen sulfide, which is a byproduct coming out from this reaction that would have a physiological role. And indeed, we set up a new methodology that we were able to detect this H2S molecule in response to the addition of the cysteine as amino acid in our cells. We compare quiescent homeostatic cells, these are from humans, and you can see that over time, they respond to cysteine, which is added here at time 0, and they generate hydrogen sulfide. However, when we check that in atherosclerotic endothelial cells isolated from patients with internal carotid artery disease, we found that this capability of fluxing the sulfur moieties is quite lost. Now, how come H2S has a physiological relevant function inside the cells? We all know that this is a very toxic gas which stops mitochondrial respiration, and of course it smells very, very bad. But the notion on the field back then was that this specific molecule, when it is produced physiologically inside the cell in low amounts continuously, then can be sensed from highly nucleophilic cysteine residues in proteins modify them with a unique modification that is called persulfidation or S-sulfhydration, and change their activity. Now, the biggest task that we had was to develop a new methodology that we can unbiased detect all these cysteines being sensitive to the hydrogen sulfide generated within the cells. And for that, we joined forces in Frankfurt, and we are able to present you today a novel sulfhydromic pipeline which combines some heavy biochemistry and mass spectrometry and also bioinformatic analysis on a single cysteine level that allow us to map the first endothelial cell sulfhydrome. Now, we used that in blood endothelial cells that were predefined as H2S high and H2S low, which are the groups that we use, 3 different groups. The first one, cultured endothelial cells with a gain and loss of function of the enzyme, so CSE high and CSE low. And we used also the CSE low cells to treat them with H2S donors. Potentially, we would have some therapeutic targets there. And of course, native human material from healthy homeostatic vasculature compared to atherosclerotic plaque-isolated endothelial cells, which have an active and inhibited enzyme. We validated everything in endothelial-specific inducible knockout mouse, and these are our results. So what you can see in this circus plot, it's nothing more than all the single cysteines being sensitive to this modification. And of course, every single one of them is highlighted here with a different color coding based on its p-value. We were able to identify about 3,400 of these residues, and some of the proteins were having more than one residues being modified. So how you end up having your best target and your most affected protein, we employed a mathematical approach that is rather used in the DNA methylation studies, in which we combine every single p-value from every single of the cysteine being sensitive to the sulfhydration event, and then identified the combined p-value, which brought us to our best target, which was the beta-3 leg of the AV beta-3 integrin. Now, this one we have validated also some more on the past years, and as well our first favorite, the RNA binding protein HUR, will serve today as 2 examples to share with you our data highlighting how these modifications impact their function and subsequently endothelial cell function. So let's start from the easiest example. This is the RNA binding protein HUR and has only one cysteine that is modified, the cysteine 13. So what our model proposes is that when this cysteine receives the modification, then HUR primarily stays in its inactive monomeric form. What happens when the enzyme is not working, as for example in early stages of inflammation and atherosclerosis development, is that the modification does not occur anymore, and now the cysteine 13 can freely interact with the second monomer, form a dimer of the RNA binding protein, and stabilize RNAs that they are linked to endothelial cell activation, for example, the E-selecting molecule, or atherosclerosis development as cathepsin S. Now, to validate that indeed that was the case, then we performed some carotid ligation studies in ApoE background mice, litterments with or without endothelial CSE expression. And what you can see in this representative micro-CT analysis is that the lumen area of the ones lacking CSE and as such having an active HUR is reduced. Now, can we target it therapeutically? To address that, we used PEROS, an H2S donor, which recapitulates what happens in the endothelial cells physiologically, very small levels of hydrogen sulfide continuously released. And we were able to see that indeed this donor is able to increase the lumen area after the carotid ligation model. So moving from the first to the second example, here things are a little bit more complicated. Now, we don't have one single cysteine being modified, but we have multiple of them. So what our model proposes is that when the beta-3 leg is not modified at all, then it tends to form disulfide bonds that they support a bent conformation of the AV beta-3 integrin, which is inactive. What happens if we increase the H2S in the microenvironment is that other nucleophilic cysteine residues that remain free from disulfide bonds get now modified. And this results in a reshuffling, of course, of electrons and finally a reshuffling of disulfide bonds, which seems to preserve an extended open and active conformation of the beta-3, AV beta-3 intergreen. Now, how to prove that? I will just share the proof of principle experiment that we performed. We used, of course, endothelial cells from atherosclerotic patients that we know that they have no sulfhydration or very low amounts of sulfhydration and an inhibited CSC, which should mirror what happens here in the bent and inactive conformation of this protein. And then we split them in two and we treat them with an H2S donor that should convert this conformation to the extended open and active. Now, the first thing that we did, of course, we added the fixed fibronectin, and with the FACS analysis, we're able to see that indeed the H2S enriched cells are having this rightward shift on binding fibronectin, indicating that certainly this beta-3, AV beta-3 intergreen is now more active. Now, to really validate the conformation, we used conformation-dependent and independent antibodies. First, independent antibody is the AP3, which binds no matter what, if the AV beta-3 intergreen is extended, open, or bended. And here you can see exactly that these two conditions do not affect surface expression of this intergreen. However, when we used conformation-dependent antibodies, then we were able to see that indeed this H2S enrichment supports the extension and opening of the AV beta-3 intergreen. And this resulted in about 7-fold increase of the intergreen activation index. Now, what is the impact of this beta-3 intergreen control? The first thing that, of course, everybody would expect is adhesion. And here we mixed these two different cell types, color-coded based on different cell trackers, and we plated them in fibronectin-coded discs. You can see that in time 0, you have pretty much similar percentage of both red and green cells, but 1 hour later, when you wash them, only the ones that they have this extended and open intergreen, and they are pre-treated with the H2S molecules can bind. Now, what does it mean for our vessels? One thing that it is lost is the mechanosensory responses. So to evaluate that, we used second-order mesenteric arteries from the mice, from littermates, and we performed flow-induced dilation studies. So what we do is we increase the flow, and this should physiologically increase the diameter of the vessel, which is the case when we have a wild-type mouse with an active CSC and an active beta-3 intergreen. However, when we don't have this process, then you can see that these vessels do not respond to flow, and we can partially rescue it with the addition of the H2S donors. Now, I think that I try to convince you that indeed this modification impacts on endothelial cell function, both mechanosensory responses and atherogenic responses. And for us, it was very important to find a way to monitor whether a patient has this desulfhydrated dysfunctional endothelium as early as possible. Now, for that, we were quite lucky, as the substrate of this pathway, cystathionine, is not consumed and it circulates specifically when endothelial CSC is not working. And here you can see high levels of circulating cystathionine in atherosclerotic patients with pronounced endothelial dysfunction, and how this negatively correlates with the capability of the vessels to respond to flow. In a larger cohort, what we found is that indeed atherosclerotic patients have high levels of circulating cystathionine and low levels of hydrogen sulfide. And of course, for us now the question is, if it is all about H2S, can we just give it back to the system and manage to resensitize the vessels of endothelial dysfunctional patients to respond to flow? So that's what we did. We used a very small cohort of patients with severe endothelial dysfunction, and we performed vascular reactivity studies on them. And you can see that these patients, after 5 minutes of constriction and then reactive hyperemia, they saw a 10% increase in their flow-induced dilation, and this doesn't change over time when we treat them with a placebo group. However, when we give back to the system what is missing and resulfhydrate some of these targets, including the beta-3 intergreen, we are able to resensitize their vessels to respond to flow. So indeed, as I showed you so far, carbon flux and sulfur flux seem to be important for endothelial cell proliferation and endothelial function. But during these studies, we have a rather unexpected finding. So we let these mice, the littermate mice with lacking CSC in the endothelium, stay for a little bit longer, and we saw that they have a rather premature lethality phenotype. So we proceed on this context, and what I will show you in the next few slides is our most recent study, which highlights that the enzyme itself has also a function that we didn't know before as a scavenging protein in the endoplasmic reticulum where it is localized that could direct transcriptional program of the endothelial cell against aging. So indeed, this enzyme is no longer present when we screened 80-year-old endothelial cells from humans compared to 20-year-old endothelial cells from humans. And as aging is linked to the reparative responses of the vascular system, we went back to the mice and generated the weak-adhering Cre-inducible mice this time, which are lacking CSC specifically in the endothelial cell. And we tested their responses to injury. So we perform a model that is called aortic regeneration model. This is relatively simple. You clamp the aorta for about 1 minute, and then you destroy the superficial endothelial cell layer. And this is how it looks when you stain for weak adherent in black, and you do an on-fast staining 4 hours after injury. Cells disappear, and there is no EDU incorporation. Now, what should happen in a normal adult mouse is that later on, 48 hours to 72 hours later, these cells bidirectionally proliferate to cover the wound. However, when you have a middle-aged mouse, 12 months of age, or an endothelial-specific inducible knockout, just 4 weeks after the deletion of the CSC, this is no longer the case. And this is because the cells seem to have a rather premature senescence phenotype. Here you can see a senescence beta-galactosidase staining in these cells of the same passage, which is much, much enriched when we delete this enzyme. And I would rather say that the most interesting finding was that they show reduced telomerase activity, which is indispensable for endothelial cell proliferation, and of course also telomere shortening or telomere abundance, as highlighted here in red with a PNA-FIS approach. So what we did next was, of course, to sequence these cells and test whether loss of the CSC indeed induced premature senescence. And these horrible heat maps here show only one thing, that the loss of the CSC in the endothelium results in a loss of all the negative regulator transcripts of senescence and an increase in most of the positive regulators of senescence. So we proceed with further analysis of these RNA sequencing results, and what we found was that the p53-mediated transcriptional control was among the ones that were much more enriched. So we thought that, okay, what is happening is that chromatin is altered and p53 can now bind in these knockout cells, but that was not the case, as the transcriptional factor motifs for the p53 remain unchanged between the different genotypes and also in young and aged humans. But what was really changing was the fact that now p53 was able to bind to its targets. And here I just share with you some examples. Now, why was that? What we identified is that in the absence of the CSC, p53 seems to be more enriched in the nucleus, where it gets acetylated on a specific lysine 120 and interacts with an acetyltransferase, which is called MOS. And here you can see these pink dots inside the nucleus highlighting the interaction of the acetyltransferase with the p53, giving us some food for thought to ask whether this lysine residue is really important for endothelial cell aging. So to answer that, we went back in culture. In this case, we used young native endothelial cells from mesenteric arteries, and we performed a lentiviral transduction on them with a non-targeted control or guide RNAs against the p53, and simultaneously overexpressed lentiviruses which were carrying a wild-type p53 or a mutant that could no longer be acetylated, and perform puromycin selection and start by evaluating the proliferative capacity of the cells very, very early. And the results were quite surprising, that indeed by deleting the p53 or not allowing the acetylation to occur in the lysine 120, we preserve and enhance the proliferative capacity of the cells. And when we culture them for more than 10 passages, which is a multi-duplication model of replicative senescence, you could rather say that indeed these cells show much reduced expression of the senescent-associated beta-galactosidase. Now, the question is, what is the effect of all this p53-mediated activation? And among others, what really was interesting for us was the hypothesis that primarily is established on the cancer field, that p53 regulates the glycolytic responses of the cells. And indeed, what we did was perform some cheap qPCR studies, and we identified that p53 binds much, much more when CSC is not there in some of these GLUT transporters. And that was functional. That was really functional. When we treated the cells with a fluorescent glucose analog, we couldn't see the glucose getting inside these endothelial specific CSC knockout cells, which have an active p53, compared to the wild-type conditions. And the seahorse analysis shows similar things, that we have reduced glycolytic event. What is the effect? The effect is that these cells show reduced ATP levels intracellularly. And I would rather say that this is one of the most remarkable ATP difference that we see in endothelial cells when we compare different conditions like proliferation, for example. So what can this mean? Of course, ATP is a very important cofactor to activate ATP-dependent proteins in complexes, including chromatin remodeling complexes. But one of its targets is the telomerase complex. And now telomerase is working properly to extend the telomeres and allow the proliferation of the cells. And this is ATP dependent, so we reasoned that this loss of ATP in these cells would impact the complexome of the telomerase, and that was exactly the case. Whatever you see here in red, it's just interacting partners of the telomerase in the wild-type situation, where you have an active CSC and a loss of function on the p53 activity, and this is gone when you delete this enzyme. So the question, of course, was, can we put back a cell-permeable ATP to take the telomerase activity as close to normal as possible, and like that allow the cells to proliferate after injury when we need to, even in aging? So this is what you can see here. Indeed, by adding a little bit of cell-permeable ATP analog, we could increase the telomerase activity, and this data indicate that all the components of the complex are present in aging, just they cannot form the complex to work properly. Now, the last thing that we had to really question is whether these are all ATP-dependent effects on the telomerase activity and proliferation are only due to the fact that telomerase is not associating with its collaborating partners. And we just knock it down, and we perform a multiduplication model, a progeria mimicking in vitro model. And what you can see here is that these third knockdown cells start losing their proliferative capacity after the fifth doubling, and up to the 10th doubling, they are already arrested. Now here, ATP is working only in cells that they still do express the telomerase, and just allows the complexome to form back. However, this is not the case if the key component is not no longer present. So what I've shown you so far is that indeed, at least in aging, the p53 can get inside the nucleus, and then here it gets acetylated in the lysine 120 because it interacts with this acetyltransferase. This results in a transcriptional reprogramming, including loss of the glycolytic flux, reduced ATP levels, and among others, impact on the telomerase activity. Now here is where the CSC comes in the game. What is really regulated p53 in aging? So it's widely known that p53 nuclear localization is regulated from ubiquitination, but at least in endothelial cells, what we identified is that the CSC, which is a very big tetrameric protein localized in the ER, traps p53 in the cytoplasm. Here you see just interaction between the recombinant p53 and its DST-CSC purified protein in silico. We perform in vitro, we perform the in silico analysis, and we identified the actual pocket within the CSC that the p53 can be trapped, mutated the proteins, and make sure that these are the regions that these two proteins interact. And what you see in these small images here is that in a young native endothelium, these two proteins do interact very close to the nucleus where exactly CSC is localized, and this interaction is lost in aging. So in a nutshell, our study highlights that loss of this interaction drives the p53 inside the nucleus to regulate aging processes. And the biggest question that we have towards translation always is, can we use the CSC as a gene therapy to trap back the transcriptional factor where it is produced in the ER and rejuvenate the endothelium? So to answer that, we used some preventive aging strategies by generating specific AAV9 mCR particles which had an S2A CSC active mutant or a dead CSC mutant, and injected them in the mice at 3 months of age, followed them up to 1 year, and what we saw is that only when CSC remains active and traps the p53 in the cytoplasm, glucose levels are high and also telomerase activity is preserved. And this increased telomerase activity is functional because when we challenge this model to proliferate with the aortic regeneration model that I showed before, only the cells that they are positive for the active mutant of the CSC can still proliferate, and this is a mark of youthfulness. Now, the last thing that we did was, of course, to try to revert aging in endothelial cells from humans by using the CSC as a gene therapy again. And what we did was to take vessels from AIDS, mesenteric vessels from AIDS, 80 years old and 20 years old human, and shortly overexpress the CSC in an adenoviral form. And what I would like you to focus here is this positive CSC cell close to the neighboring AIDS negative cell. While the negative cell saw extensive expression of the p53, including into the nucleus, the positive cells show such a reduction of the p53 expression and nuclear localization, and then when you FACS sort these cells, this telomerase activity is relatively high. And this brings me to the end, close to the end. So with these 3 studies, I would rather hope that I convince you that this single amino acid and its pathway have relatively important functions for maintaining chromatin control, endothelial cell homeostasis through post-translational modifications, and of course, an unexpected role of the enzyme itself in aging. And I would rather end by saying that indeed cysteine can drive the destiny of the endothelial cells, and these cysteinolytic high cells are rather metabolically primed, that they can proliferate when they need to, and they have resistance on exhibiting senescent and transdifferentiating characteristics. And if we identify exactly the mechanisms that this enzyme is selectively regulated in some clusters of endothelial cells, we could potentially have some novel molecular targets for AIDS-related vascular diseases. And I think that with that, it's time to give my thanks to the Institute of Vascular Signaling, who is hosting me all these years, and in particular, Professor Fleming, which I cannot see at the moment. Thank you so much for the great mentoring all this year. All the members of the Institute of Vascular Signaling, and my long-lasting collaborator, June, who is also, we are sharing the authorships in this work. My own team, a group of very motivated people, and Maria is here with us today, so please feel free tomorrow to join her poster session and learn some more about her work. Our big family in Frankfurt and our brother institute, as well as our national and international collaborators and our funding bodies, and thank you all for your attention.
Thank you so much, Dr. Biblis, for a wonderful talk showcasing beautiful data and a variety of techniques and approaches. So in the interests of time, I don't think we have any time for questions because we need to give everybody a break before the poster session. But perhaps if anyone has any questions, they can approach you now after the talk. So it just remains for me to congratulate you again on this prize and award, and to present the prize and award to you on behalf of the Physiological Society.
Thank you. We can take a photo, I guess. No.
I think so. Are we getting photographs? Okay, right. Okay. Thank you, everybody.